Okay,
Hi
Ella.
everyone, welcome and thank you for joining this first UNA webinar about AMR. It's great to have you with us. I'm Caroline Horner, the scientific lead at UNA Health, and I'll just spend a few minutes giving you a quick overview about the AMR kits available from NG Biotech. So currently there are 4 cassettes available: Carba-5, which detects carbapenemases, which is what we'll hear most about today. There are 2 kits that detect the CTXM family of ESBL enzymes, and there's a kit to detect the mobile colistin resistance. So in terms of a general overview about the cassettes, the kits have been developed by ISO accredited labs in France using quality biological materials, which leads to the development of quality kits. All 4 cassettes are CE marked. They have an excellent sensitivity and specificity when compared with molecular methods, but they're less expensive than PCR, and you don't need any additional pieces of kit apart from a vortex. So they're simple to use, rapid to set up, and you get your results within 15 minutes. They are easy to interpret, and their kits have strong test bands, so you can be confident when you're reading the results. So this slide just shows a Carba-5 cassette on the left, the sample well, and the well where you get all your results. In the middle is the procedure, the test procedure. So you add some buffer to a microtube. You have bacterial colonies grown on selective or non-selective media. You add that to the lysis buffer vortex and take 100 microliters and add it to the cassette. The bottom image here shows the Carba-5 results and the strength of the bands for each of the carbapenemase families. So one of the advantages and the strengths of the Carba-5 kit is the range of variants that are detected from the big 5 families of carbapenemases, which is shown on the screen now. And it's a single cassette that detects all these variants. So not only is that better for your workflow, but it's also good in terms of plastic waste and your disposables as well. The kit can be used on Enterobacterales and Pseudomonas aeruginosa. All the kits can be used direct from positive blood cultures if that is what fits for your workflow. So in this step, there's an additional blood prep kit, and you add 1 ml of the blood culture into a tube, give it a wash, end up with your cells at the bottom of the tube, and add the buffer as you would, and then add it to the kit. So another simple, quick procedure for you to get a result. The other kits in the range, as I mentioned, are 2 for CTXM. So the single CTXM detects group 1 CTXM family, which contains CTXM15, the most common CTXM. The CTXM double M is a multi detector for all 5 of the CTXM families of ESBLs. And the final kit that's available at the moment detects the mobile colistin resistance encoded by the NCL1 genes. So another important resistance mechanism. So that's a quick run through of the 4 kits that are available. While you're watching the webinar, if you'd like to add some comments about the kits or any questions you have, as Peter mentioned, into the question and answer session, we can answer those. We're particularly interested to find out if a direct detection cassette, so direct from clinical samples, would be of interest to your lab and your clinicians, and just in general what your lab struggles with in terms of resistance detection, if there's anything that we can help you with there. So that's it from me. Please get in touch with Una via the email or on the website, and I'll hand back to Peter to introduce the next speaker. Thank you very much.
Hi, so my name's Lydia and I'm here with Carrie-Anne, and we're both biomedical scientists from Wiston Hospital. So we're just going to talk to you today just how we would validate a method to bring into use into the lab, of how we would validate this kit for use on patient samples. So just a little bit about our trust. So St Helens and Knowsley Teaching Hospital is part of a wider network including Southport and Ormskirk hospitals. We process samples from both trusts, and we've done this since 2015 as part of the merge. We also have a specialist burns department and specialist spinal injuries unit as well. So just a little about our microbiology lab. So we process approximately 720,000 samples a year. That's about 60,000 samples a month, including 20,000 COVID samples. We are 24/7. We're only one of the few microbiology laboratories across England that actually run 24/7, and we read plates throughout the night as well as urgent samples. We process a range of sample types, including urines, blood cultures, CSFs, a range of urgent samples, including tissues and fluids, and we do epidemiology screening, which is where the CPA and ESBL kits come into. So our epidemiology screening, as I just said, runs throughout the night. We have 4 reads across 24 hours, and we look for CPA, MRSA, VRE, ESBL, and Linezolid resistant staphs. So that was just about the trust before, and then just the range of samples we process, and just a bit about our lab. And then this was just the slide I was on then about our epidemiology screen. So we are quite lucky in our area. We don't actually get that many CPE positive samples. We have had 22 over the past year. However, that is just on our screening bench, and that's not any incidental findings, which we do pick up on other benches. So our current method, we use the Cepheid Carba-5 kit, and everyone knows they're obviously a well-known brand, and we've used these for a while now. It's sensitive, reliable, and it's good that our results transfer to our LIMS system. However, they are costly, and there is times where we have to pause for maintenance and downtime, and especially at the moment we're using so many Cepheid kits for our rapid COVID samples, so we do need to free up that machine a little bit. So we've considered changing our method for CPE detection. So this is just a few questions that we need to ask ourselves. So what is the need to change the method? Is it cost, which at the moment it is? We could save a large amount of money by switching to the NG Biotech kits. Will the reason to change positively impact patient care? If it can reduce turnaround times, then yes, definitely. And does the alternative provide a cost saving, a SIP saving, which is what we want? And is there training involved? And there's only a minimal training involved, which is really good. So this is a potential new method by using the Carba-5 assay. They're easy to use and implement, minimal trainings required. We can reduce our turnaround times to 15 minutes rather than the 50 minutes.
So we did actually trial this test in-house so just before the pandemic. So we did a trial against the Cepheid PCR kit that we currently use, as Lydia said. And during the trial, the isolates that we used known quality control strains and clinical isolates. And within the clinical isolates, we just tried to represent like a diverse range of Carbapenemases genes from the big 5, which we test for with the Cepheid now. So during the evaluation, we followed the manufacturer's instructions for both testing methods. So we took a bacterial colony from overnight culture, and we actually tried to use a variety of media. So all the isolates that we used, we grew up on 5 different agars. So we used the CPE media, everything from E&O, so the CPE media that we normally use, and it's the most common place that we would isolate a colony from. Our UTI chrome, normal blood agar, CLED, and then we used our sensitivity agar as well. So with the Cepheid PCR, the results are quite easy interpreted by the machine itself, and you get positive or negative. For the Biotech Carba-5, we performed the test in 15 minutes, and the positive result was given to the definitive pink line. So the isolates that we used 60 isolates that we previously had in the lab. 30 of these were CPE negative, and then we used a range from the different from the 5 genes. So we had 10 up to 48 NDMs, 4 VIMs, 9 KPCs, and 1 IMP positive. And some of the isolates had more than 1 gene. So the outcomes of the evaluation were really positive. We've seen 100% correlation between the Carba-5 Biotech kit and the Cepheid PCR when it was taken off 4 of our agars. We did have some discrepancies from CLED, but it still had a 91.7% correlation for positive and negative results, which is still really good. We had five samples, which you can see in the table, where the biotech was negative and PCR was positive, or it didn't detect one of the genes. But this wouldn't put us off using the methods. We just wouldn't really take it off CLED anyway. It's just good to be aware of that sort of limitation. But we had 100% correlation from all our other medias. So we found that the test was really easy to perform. There was minimal training needed, didn't really need any other equipment, and the tests were really easy to read, which we had concerns about before we started doing the test. We were worried that we would have transcription errors, or because it's you're reading a line on a test, if it would be faint lines, but we found apostrophe was really obvious. We had a really good pink line. So in our actual evaluation now, we've completed all our documents for UCAS and we're just waiting our consultant's approval. So we've got really good confidence within the lab in using the test. So we just need to get our medics on board. So our plan is to use the test alongside the CEPID for another month or so now that the pandemic's sort of quietened down slightly, and get some more data for us to be able to stop using the CEPID and only use this for confirmations. And just a little slide about further work. So one of our undergraduate students, Anna, she's just done a project on ESBL detection. So she used the Biotech CTXM multi-kit and she compared it against a range of other tests that we use. So against our Biotech, ESBL Roscoe kit and disk synergy testing. And she found really positive results with that for ESBLs that are positive for CTXM. And she's actually working with Una Health to do an article for the website, so just watch this space on that one. Thanks for listening, and if you've got any questions, me and Lydia are more than happy to answer them, but if you just pop them in the chat, I think they'll be fed back to us at the end.
So we've been using the Carba-5 lateral flow for a number of years now. So I'm going to summarise our results and our method of how we use the lateral flow within our screening algorithm, and then some of the little changes that we're going to make. So to put you into context, Barts Health is one of the largest NHS trusts in England, and we're actually now expanding and becoming East and South East London pathology partnership. So we treat now more than 2.5 million patients. And what's important when it comes to our carbapenemase screening is a significant proportion of our patients are from Bangladesh, Pakistan and India, which according to the PHE toolkit puts them at higher risk of having a CP compared to other members of the population. So the reason we wanted to implement the Carba-5 lateral flow initially is because we had an OXA48 outbreak on one of our surgical wards, and we needed a quick method to detect the patients that were positive versus those that were negative. And as part of this, one of our BMSs conducted a points prevalence study as part of her professional doctorate and identified the renal groups and also care of the elderly as having the highest burden of carbapenemases. What was more interesting was when we looked at the carbapenemases detected within our trust, we found actually the country of birth was not such a high risk factor. So even though we have such a high proportion of high risk patients, we weren't actually seeing a higher proportion of carbapenemases within them. So our screening method, we culture all of our swabs onto the M-Supercarba media, and then we will identify any growth by MALDI-TOF. So our screening strategy is purely for CPE. We don't look for any other organisms at present unless there's a specific outbreak. So anything that looks like it's a Enterobacteriales then has a sensitivity test, which has a number of antibiotics, but the ones that we tend to look at are temacillin, piperacillin, tazobactam, meropenem and ertapenem. And it's a two-prong attack. So isolates that are temacillin resistant and piptazo resistant, but meropenem susceptible, these are our presumptive OXA48 producers. So the problem with this particular antibiogram is it also selects for ESBL producers. And then we also look at isolates which are meropenem and ertapenem resistant, because regardless of what other resistances they have, these tend to be the other carbapenemases that we might pick up. So any isolates matching these two prongs of the algorithm will get a Carba-5 test. And I've listed here the alleles, but I appreciate has probably changed more recently. So between March 2019 and March 2021, we've tested nearly 13,000 rectal swabs, and we've detected carbapenem-resistant organism in just under 2%. And of these, we've detected 1 CPE in the majority of our samples, and a small proportion will have 2. And what we generally find, as you would expect, the majority of CPEs have just 1 enzyme, but we do have a small number that will have multiple. So just as our outbreak when predicted, OXA48 is our biggest protagonist in the trust, followed by NDM. And then we've also detected a handful of KPCs and IMPs, and the biggest combination tends to be OXA48 with NDM. And we've also detected 1 IMP with NDM. This other group, I should point out, when we first started screening, anything that looked like that was carbapenem resistant but Carba-5 negative, we would send to the reference laboratory just in case we were missing any other carbapenemases. And apart from 1 guess enzyme, which we detected, everything else was coming up as carbapenemase negative. So likely these were the query ESBL producers. So now what we do, rather than automatically sending these isolates, we only send them on request, because generally they're not carbapenemases. What we also find is the majority of our CPEs tend to be E. coli and Klebsiella pneumoniae, as you would kind of expect from the UK epidemiology. And do I have anything else? I don't have anything else. It's a nice quick presentation. So one thing which we are changing, we've got a couple of changes coming up as part of our new pathology partnership. So we will no longer be using ertapenem as part of our screening tool, because realistically this doesn't really help us when it comes to detecting the carbapenemase producers. Meropenem is the better indicator. And also what we're now going to be doing is more screening of Pseudomonas isolates, which are linked to outbreaks, but they will have a different algorithm. So we will only follow up those when they are ceftazidime, piperacillin tazobactam, meropenem, and ceftolozane tazobactam resistant, and otherwise they will follow the same procedure. So as Peter said, any questions, please pop into the Q&A and we will answer them later.
My name is Joanne Bullivant and I work at Sheffield Teaching Hospitals. I'd just like to thank Una for inviting me to speak today, and I'm just going to go through a bit about how we use the Carba-5 in Sheffield. Next slide.
Okay.
So we have used LFTs for a few years now in Sheffield, but we had to switch to Carba-5 as we identified an issue of poor sensitivity for the detection of NDM isolates. In Sheffield, we have a large spinal injuries unit receiving patients from across the country. And although we do not have a massive problem with CP isolates, we do see a few NDMs on the spinal injuries unit. With the LFT we were using, we were seeing quite a few negative results, like as in false negatives. And so we were referring a lot of results to reference lab for confirmation, and they were returning back as NDMs. So as a result of this, we looked at alternative kits and started investigating using the Carba-5. Right, so sorry about that. As I was saying, we were having identified some poor sensitivity of our NDM results, which was not ideal in how we were using the lateral flow. So we were sourcing an alternative, and as can be seen in the picture below, this is the same isolate run on the Carba-5 and the competitors, and you can see a line at the NDM result, which is negative in the other cassettes. Next slide, please.
Okay.
So sorry, just going through. So at Sheffield Teaching Hospitals, we are lucky that we have several methods for detecting CPEs, and with each method we use to complement each other. So for our screening for CPEs, we actually are lucky enough that we are able to borrow media. So we were able to validate a CLED agar, including, when initially we used ertapenem, but under the toolkit we transferred that to 0.125 meropenem. So this plate here is actually cleared with 0.125 milligrams per litre meropenem in it. So this is for our CPE screens, and so anything that grows on this plate, the following day we will get a MALDI-TOF ID, and if it is an Enterobacteriaceae, we would consider this a potential CPE. The other way we screen isolates is actually all clinical isolates. We actually use breakpoint agar incorporation for sensitivity testing, and all our clinical isolates are screened. So not only in our breakpoint set do we have meropenem at the breakpoint concentration of 2, we also include another plate with meropenem at 0.125. And again, all our Enterobacteriaceae that grow on this plate, we will MALDI-TOF direct from these spots and get an ID. And if the ID is an Enterobacteriaceae, we will go on to do further work. How the Carba-5 comes into play from here is if this is an inpatient pathogen, we will directly do a Carba-5 at day 1. And we will do this from our screening isolates and from our incidental clinical isolates that turn out to be meropenem 0.125 resistant. This information, if positive, is fed directly to the infection prevention control team and the medics, and they can implement procedures at day 1 rather than wait for further results on day 2. So in addition to this, any isolates that are then positive for with the Carba-5, we still use Roscoe discs because we feel that these capture all results. Prior to the Carba-5 or a lateral flow test, we would have done full Roscoe discs overnight and waited for the results of these. Obviously, these can only tell you that you have an MBL and can't differentiate between NDM, VIM and IMP, which the lateral flow can. In addition to this, we also have the Kefi PCR. Currently, we use this just for screening isolates that we require a rapid turnaround, so direct from swab, for example, transplant patients. But we can use this also for isolates if we've got conflicting results between some of our tests, which I'll go on to talk about on the next slide. And obviously, we also have the medic suspicion, and if they feel that an isolate, like for risk factors from travel or lots of inter-hospital transfers, we will perform a Carba-5 on day 1 at request. So as you can see, we are fortunate to have a whole range of tests available to detect CPEs. The Carba-5 offers a very quick result, which enables infection prevention precautions to be put in place on day 1. And if we have a negative Carba-5 result, we will still continue to do our overnight cultures on Roscoe discs, because this can use to back up that these may be hyperproducing abscesses within your Class C Carbapenemases, which are obviously not true CPEs, but then we can tie all those results together on day 2. Next slide, please. So based on a bit of our algorithm, I just thought I'd talk about a few interesting cases where the Carba-5 has given us some positive results and how we've been able to use them. First one is multiple mechanism detection. Although rare, we have had a few isolates that have dual mechanisms present. This obviously makes a little difference in terms of IPC, but it is important in cases of outbreaks if we're trying to monitor which plasmid has been transferred. If we relied, as I said before, solely on the Roscoe discs, we probably wouldn't have picked up that we had an NDM and an IMP because we've just had them as MBLs. So the Carba-5 allowed us to pick up both of these results. Interesting, because these were dual infections, we did put these on the Kefi just for confirmation, and the Kefi picked up both these mechanisms as well. Second thing to talk about is we have had some tricky isolates and conflicting results. We've had a Carba-5 negative result on day 1, and then we will have set up the Roscoe disc as part of our algorithm to confirm that result, expecting to get AMPC and porin loss or ESBL and porin loss, and that they've demonstrated a KPC. Now, these isolates have been in Enterobacter species, so of known intrinsic AMPCs. So they're always a bit dubious anyway, because the AMR was literally just above 0.125. However, the Roscoe discs were demonstrating KPC, showing that the cloxacillin was not being potentiated appropriately to give a big enough difference in the zone sizes. So these caused us a little bit of concern, because at the time we weren't sure whether it was the Carba-5 that was wrong or the Roscoe discs that were wrong. So these isolates, we went on to perform a care feed, and they came back negative as well. So in this case, it's actually the Roscoe discs that were giving us a false positive, but the Carba-5 was the correct result. And the last one I want to talk about was just one that shocked all the medical staff, was we had an Acinetobacter that was imp positive. This was from a clinical specimen, so not a screening swab, and it was at the Children's Hospital. So actually on day one with Acinetobacters, we wouldn't routinely perform a Carba-5. We'd wait until we got some sensitivities on it, and if the medics thought that there was potential that it may have a CP, then we probably would do a Carba-5 and send to the reference lab. We don't do a lot with Acinetobacters. However, in this case, the medic had a suspicion that she wasn't quite happy with, and seen that we'd thrown an Acinetobacter, and rang the lab and asked for a Carba-5 straight away. She said, I expect it to be negative, but I just feel like it's something we need to do. And we ran it, and it was an imp positive. Now, this patient is a complex patient under the care of multiple disciplines, and was actually in hospital in a bay. If the medic hadn't requested that result on day one, we hadn't been able to give that result, that patient was sat on the bay for a further 24 hours, increasing the number of contacts in the bay. It actually opened up a huge infection control measures were put in. There was a lot of contact tracing. Before this patient had visited, was under multiple care pathways, so they all had to be contacted and screening put in place. So a great positive for the Carba-5 is if we hadn't had that result, it probably would have been another 48 hours before we'd confirm that this had an imp, so that would have increased the number of contacts and increased a bigger headache for the infection control team. As a result, this patient was moved to a side room with immediate effect, and all the appropriate measures put in place. So next slide. Really, that's it from me. Sorry about the technical issues. I mean, if you have any questions, please pop them in the discussion and I'll answer them as soon as I can.
Hello everyone, I'm Francis Youngblood. I'm the laboratory manager for microbiology and virology at Great Ormond Street Hospital, and my presentation is on detection of CPE in a specialist tertiary paediatric hospital. So I guess you've all heard about Great Ormond Street Hospital, and so we are a specialist referral hospital. So compared to a lot of other trusts, we don't get as many patients. We get about 250,000 patients per year. It's a high international population that we also receive too, because again, we do kind of specialist tests and procedures such as bone marrow transplant, etc. We also receive referrals from district general hospitals. So as a kind of a referral site or specialist tertiary hospital, we don't actually have an accident emergency. We just get patients referred to us for special treatment. So looking specifically on our enteric bench, we get about 500 stools specimens a week, which may not seem much compared to other places. So I guess, what do we do on our enteric bench? Well, basically, it acts to look for your gastrointestinal pathogens, but in addition to that, we're also looking for resistance. So we look for gentamicin resistance, ESBL AMP-C producers, querying CPE, and resistance to cephalosporins and any other kind of resistance that may be present, such as VRE. Again, there can be outbreaks on wards, but why the enteric bench acts more as a screening bench is because of our patient population. So again, we've got patients that are immunosuppressed, heme-oncology patients, where basically anything that they carry in their gut puts them at more risk of getting sepsis, basically. So it's quite crucial that we kind of monitor carriage and do this screening to know what they have actually got in their gut. So in about 2016, we actually had an incident where we had a patient referred to us from another trust, and we actually isolated an OXA48 that we actually had missed in the laboratory. And actually, it was only because of communication between the district general hospital that we found that actually they had this pathogen, but we hadn't detected it. And that's actually quite a critical issue in terms of infection prevention control, which other patients were in contact, etc. So we came up with an algorithm. So this was the one that we had previously. This was about 2015, 2016, and it's quite complex, but I think we learned our lesson that actually we don't want to actually miss anything. And so you can see this is what they basically have on the bench in the laboratory. So we look at our coliform, and if it's being followed up, you carry out resistance testing, sensitivity testing, and follow this algorithm. So if you just look, I think it will be on to your left side, it says about possible CPE, and so we would ID, save the isolate in case we need to do any further work. We look at the sensitivities for meropenem, terpenem, tazobactam, and temacillin, and also do MICs to confirm. And at the time we were using the Roscoe kit. So what we found in terms of our patients is that actually we got quite a few false negatives results. So we'd always send to the reference laboratory to actually carry it out. So I'm just going to go to a case study. So this is just a case study using this old algorithm, and it was actually looking at a bone marrow transplant patient that was in a ward. So actually they had a screening sample come. So for the bone marrow transplant patients, they have one stool that is sent weekly to monitor what they're carrying in their gut, so that we, if we find something resistant, we know what they're carrying, and if they become septic quickly, we can treat them immediately. So because there was a query CPE on the enteric bench, it was actually followed up, and we needed to wait for the reference laboratory result in order to treat the patient and actually reduce the infection prevention control precautions. So sending to the reference lab, there's obviously a delay. There's a delay in getting the result, but in terms of other management for patients generally, so you have problems with your infection prevention control precautions because you can't actually relax them. It also affects antimicrobial stewardship depending on what is happening with the patient. So we decided to look at other tests, and so we decided to look at using a rapid lateral flow. Initially we did use the kind of Chorus, but then we moved, because it only had the resist 4 at the time we were using it, we needed the whole 5 in order to target the 5. So this is the algorithm we currently use at the moment. So you've got your kind of sensitivity reading. As you can see, the red box to the left, which says possible CPE, what we've included in there now is the Carba-5. And generally, if you've got a positive, we're happy with that, but it's a negative, we don't want to miss anything. We have some kind of further follow-up tests which we're kind of looking to investigate. So implementation of the Carba-5, well, mainly the reason we did it is to improve kind of patient care, because you get the results so much more quickly. It's about 15 minutes in terms of sending something to the reference laboratory, and actually the sensitivity and specificity, as you can see here, is pretty good. So I'm just going to show another case study, which is actually looking at a different patient with a new algorithm. So this was a patient that was from an intensive care unit, and they again had a sample brought to the laboratory, which was for screening. And so it was query CPE in terms of the follow-up, and actually using this new algorithm, we actually got a result more quickly. And so actually that improved the infection prevention control intervention. And so we were able to release them more quickly. Okay, so we are now looking at actually introducing the Carba-5 for Pseudomonas species. We're currently using the MBL strips, and we get a high number of false positives. So we send them to the ref lab and we're coming back. So one of our BSc students has actually done a validation on this, and we're looking to bring that on in-house. So I'm just going slightly off topic, not much, but it's all, I mean, the whole reason of rapid diagnostic testing is to improve antimicrobial resistance. And so there's a project currently going on here and part of UCL called Precision AMR. There's been a lot of funding from the government, so it's about 3.3 million, and it's across UCL, GOSH and UCL to accelerate development in new tools for patients. So it's diagnosing antibiotic resistant infections faster to enable early treatment. And also treatment with the right dose of antibiotic combination of drugs. Also rapidly confirming that no antibiotic resistant infection is present, so that unnecessary treatment can be stopped. So it's quite significant, but where I think the Carba-5 sits in this is that actually, as well as the drive to improve antimicrobial resistance, there's also a drive to improve savings and costs. I think we all are aware that the NHS is full of drives and incentives to try and save costs. So this is some work that we have done previously in 2019, which was presented at ASM, and it's actually looking at how you can change your workflow. So we have been looking at testing isolates, but something that we're looking to in the future with AMR Precision is actually testing of direct clinical specimens, so actual stools, to see actually, can we get that result more quickly? So if there's a clinical indication that the patient does have a CPE, can we test their stool directly? And the reason why we use stools is because it's a pediatric population. Using a rectal swab, there's bleeding. So for our patients, we tend to use stool specimens. So this is just a bit of work that we did this with Chorus, but we are going to look with AMR Precision at the Carba-5 in order to capture more targets. But actually, using direct samples with the lateral flow, I think there's some big savings in terms of reducing cross transmission, improving patient isolation, and also improving antimicrobial stewardship. So that is it from me. If you have any questions, please pass them on. Thank you.
So hello everybody. So I'm Boris from NG Biotech. So before to go a bit in the presentation, let me introduce the company. So basically, NG Biotech is a French company. We're based in Brittany region, in the region of Rennes, or near Mont Saint-Michel, for the people who knows a bit of France and who visited already. So we were 30 people company, and let's say thanks to COVID, we grew up a lot, and now we are around 120 people working for the company. So we specialize in 3 different range of product. Let's say first of all, of course, is AMR, so antimicrobial resistance, but also we produce pregnancy tests and at the moment some COVID-19 testing systems. In my presentation, very quick presentation, I would like to focus a bit more on what will be the next step for NG Biotech related to antimicrobial resistance. So first of all, we try to expand our sample preparation protocol. So we know that our test is pretty rapid to get a result, 15 minutes. However, there is all this culture step before the test that we want to reduce in terms of time while keeping the same sensitivity. So as you have seen before, we have this blood culture prep for Carba-5, and we try to investigate with which kind of test we can use it, and to try to maximize the use of the blood culture prep to reduce the culture time. And also we do some investigation on the fact to use urine, fecal or blood positive samples directly with the test without the culture time, in order to have a very, very fast response, and of course to save time, but also to save the let's say the patient situation, and to make sure that they get the good treatment at a good time. What we also try to investigate is to recommend our test in parallel with the Malditoff that will characterize the bacteria that you are in presence. So we are not running in any competition with the Malditoff, but it's more like a parallel technology that we believe is useful to categorize the bacteria and to have the maximum information before to start the treatment. And of course, we are also working on, let's say, secret projects, let's say something like that. Obviously, additional rapid tests to complement our current offering. So I cannot go too much into detail for today, but as long as we have more staff, and obviously more staff in the R&D, we are working on so many different projects, whatever it is for human health and maybe some other categories. So yeah. Always committed to find solutions to detect antimicrobial resistance and to be as fast as possible to get a result in order to get a good treatment for the patients. So that's a very short presentation, but you are always welcome to contact either our distributor in UK, UniHealth, which is obviously very active, as we are having a webinar today. But also, if you need more information, you can also contact us and NG Biotech, which is located in Ypres. So you have here our let's say phone number and also website in order to contact us. We're also very active on social networks, including LinkedIn and Twitter and YouTube for videos. So please visit us also on these platforms in order to get more information on our product.